Welcome everybody to the sixth and final webinar of the EXPBIO project. EXPBIO is funded by the EU Interreg North Sea program, and the program purpose is to help restore communities, the habitats around degraded habitats, make them more natural again. And one approach of that is, of course, regional seed mixes, as many of you know from the previous webinars. And in these talks, it was mainly about how to make regional seed mixes, how to use them in the environment. And today's speaker is addressing the issue in a bit different way, because actually there were some questions in the previous webinars where it was where the question was around the topic of how to propagate the seed. And Malte Conrady is an expert. He has worked for quite some time on what does it really do with plants when they are cultivated in the fields and then brought back into the natural environment. And these are important topics to consider when using seed mixes. And therefore, I'm really grateful to you, Malte, that you agreed to present your information, your results, and your insights into regional seed mixes, which certainly give a different angle to the topic. So,
with pleasure.
With that, I hand over to you, Malte.
Thank you very much. So, first, the part where I share my screen and hope that everything works. So, let's try this really quick. Can you see the first slide of my presentation?
Looks perfect.
Awesome. I can't see myself, but no, okay, doesn't matter. Okay, so thank you very much for the nice introductions. I want to talk about the importance of evolution in plants cultivated for ecosystem restoration. At the beginning, I want to emphasize the enormous scale of the need for restoration, as we already heard, I guess. Currently, we are in the UN Decade on Ecosystem Restoration. That is in part because nearly 50% of the world's ecosystem are degraded. The area that needs to be restored worldwide with 2 billion hectares is gigantic, and it is estimated that the restoration business is worth around $1.6 trillion annually, at least in 2008, which is by now, I guess, even more. So there's a huge number of restoration projects going on at the moment. And as seen in these examples, some of them are enormous in scale, where a huge amount of seed is needed. For example, in Australia, there's, can I use laser pointer? Yes. For example, in Australia, there's 100,000 hectares of mining disturbance that needs to be restored, or in the United States, over 9,000 hectares of tallgrass prairie needs to be restored. So, as seeds are often the starting point for restoration projects, we need to know, where should the seeds come from? How do we get them? Especially if we keep in mind that often, even after re-establishing favorable conditions like restoring the abiotic factors, the target species often can't reach the restoration site because of limited dispersal in an ever disturbed environment. We could go to the nearest hardware store and just buy seeds, basically from cultivars, but these seeds would be basically the same, genetically very similar. These seeds would not be locally adapted, but at least they would be cheap and available. But what do I mean with locally adapted? Just very briefly, this is a picture of Arabidopsis thaliana. It is all the same species. It just comes from different regions. So plants can be locally different or locally adapted to abiotic and biotic, the abiotic and biotic environment. We could show that in an experiment that pollinators reacted differently to plant communities of the same species coming from different regions. It was mainly driven by the time the plants flowered. So here we used seeds from different regions grew them all in one experiment here in Münster, and we could see that seeds or plants coming from different regions had an impact on the pollinator community. So in order to restore, we want plants that are kind of locally adapted. And as of the year 2020, as previous presenter Walter Blicker has already told you, it is even mandatory for grassland restoration to use local seeds. The gold standard for locally adapted plant material is local provenancing, or using a hay transfer or green hay transfer, meaning mowing a pristine area that is right next to a disturbed area or degraded area, and just transporting all the propagules and seed that way. This would be really great if we could do that all the time, but it is not possible on a large scale. And if we overharvest the donor side, it could be even bad for the damaging for the donor side. So therefore, commercial production of regional wild plant seed material is being used more and more to cover the demand. In theory, these seeds are then cheap or cheaper and available and locally adapted. This is a system that Walter Blicker has already explained. So just very briefly, in Germany is divided into 22 regions. And within these regions, seeds are collected from multiple sources, mixed together. These are then called the F0 seeds. These seeds are then propagated in monoculture for up to 5 generations, as there is concern that the plants might adapt to the cultivation conditions. Seeds, yeah, let's leave it at that. So what we already can see is that the conditions from the natural conditions, the natural environment is very different from the fields, the cultivation fields. This could lead to selection or bottlenecks or evolution, if you will. For example, some examples. The inter and intra-specific competition is completely different. Now the plants fight against themselves, against the same species. The water and nutrient regime is completely different. And maybe a big one, the harvesting time, because under propagation situations, only the seeds that are ripe and still on the plant at the exact moment of harvest can be collected and therefore can contribute to the gene pool of the next generations. Everything else is out. So we now know that we need first huge quantity of seed. Second, seeds or the resulting plant should not be different from the wild collection seeds because of local adaptation. And third, the genetic diversity should be high so that the plants are still able to react to new stressors like global change or climate change. So this talk is now divided into 3 chapters where I want to answer if there is evolution and if there's change in these plants. First, are plants in later generation genetically different? Second, are plants in later generations different in morphology and phenology? And lastly, do cultivated plants react differently to stress than plants grown from wild collected seeds? So let's jump into the first chapter and the first paper. Here we wanted to know if there are genomic changes from natural population to the propagation in monoculture. For that, we established an experiment on the roof here of the Institute for Landscape Ecology in Münster, all the way around the roof. We had 19 different species from 2 seed producers, one from Germany and one from Austria. It's up to 5 generations and in total about 1,800 plants. We were very lucky to get the seeds from the seed producers from all generations. So from F0, F1, F2, and so on, up to F5 in some cases. So what that means is basically we have the great grandmother next to the great-great-grandmother, and so on up to the daughter, if you will. So we could grow them under the same conditions in the same year next to each other, and then observe changes in genetics or in morphology or phenology. We had for each of these accessions 18 replicates. For the genomic analysis, I will keep this part very short. We collected seeds, freeze-dried them, and then used single nucleotide polymorphism, or SNPs, SNPs, SNP markers, by applying genotyping by sequencing approach using ddRADSEq with pooled samples. So basically we had neutral markers, or markers where we don't know if they are neutral or selective. We have just over the whole genome random SNPs. With that, now we could calculate the fixation index, or the FST. This is a measure of genetic differentiation obtained by comparing the genetic variation within and between populations. So what we did here is we calculated the FST between the F0 seeds and the F1 seeds, F0 and F2, F0 and F3, and so on. And then we put all cultivation lineages together, standard it per cultivation lineages, and we saw a gradual and significant change over the number of generations spent in cultivation. So the fixation index, or the FST, changed significantly, very small, but significantly with a number of generations. This sounds concerning, but as populations almost always change from between one population to another, or from one generation to the next generation, we need to add context to see how big the change really is. Luckily, we had five species from multiple seed sources. With that, we could compare the difference by distance and the difference caused by cultivation. So meaning we had, I don't know exactly, for example, seeds from region 7 and from region 4, for example, and then could compare the genetic difference between regions, and then the genetic difference could calculate this difference, and then calculate the difference from the same subset of these 5 species from F0, so the wild collected seeds, to the last generation in cultivation. And by comparing this, we could then see that the FST values are way smaller in the wild versus cultivated. So the change caused by cultivation is way smaller than the change by, let's say, isolation by distance. Maybe to better illustrate this, this is not in the paper, but just to better visualize this, I made a DCA of some species, exemplary species. Here we can see that all the generations of a species within a region are closer together, or let's see it like this, are closer together than the generations in cultivation. So we see that these data points, all representing one generation, are still really close together, even though they are being cultivated. These are multiple generations, but the difference between them is rather low compared to the difference between the same species, Silena vulgaris, from a different region. So we see, yes, the cultivation changed the seeds, but way less so than this propagation. Another concern was that the genetic diversity declines by this propagation process, as there are lots of potential bottlenecks. So we calculated the expected heterozygosity. It is a measure of genetic diversity within a population. It represents the probability that 2 random alleles, randomly chosen alleles from a population, are different. A reduction in expected heterozygosity over generations suggests genetic erosion, which could affect the adaptability of restored plants. Maintaining a high expected heterozygosity is desirable to prevent evolutionary, preserve evolutionary potential and resilience in restored ecosystem. But what we can see is that the genetic diversity is not declining. It is even increasing. And we can see it is not increasing linearly, but it is increasing from the F0 seeds to the first generation in cultivation, and then more or less stays the same. Here in the last generation, we only have small, a few number of samples, so I would say it stays more or less the same. The genetic diversity, it increases from the F0 seeds to the first generation in cultivation. This is probably because of the initial mixing of the source population within one region. To better illustrate this, we were asking ourselves, why do some species change more than others? Or is there a way to predict which species will change more? And then we can better understand this mixing process maybe. So, for example, we looked at the mating system, where we saw that self-compatible species changed more, meaning a stronger increase in genetic diversity, than self-incompatible species. I try to explain it like this. Individuals that grow in close proximity under natural conditions have a higher chance of being closely related, like these three here, these four here, which enhances mating between relatives due to limited distance of pollen dispersal. However, during all steps of seed propagation, seeds from individual plants are mixed and neighbors are unlikely to be close relatives, even more so if multiple source populations are included. This, together with high population densities in the field, promotes outcrossing. So this increased outcrossing due to mixing increases the genetic diversity, which is a good thing. And we see that in self-compatible species, the increase in FST, or it's more difference in self-compatible species because of this increased mixing. So let's go to the next chapter. Here we wanted to know, are plants in later generations different in morphology and phenology? As the experimental setup was the same as before, I will almost directly jump into the results. Just as a reminder, we had the different generations, the great grandmother, mother, daughter, and so on, at the same time, it is under the same condition next to each other. And then we measured biomass, height, number of inflorescence, so number of flowers, and start of flowering. What we saw are the results here. We see that the different cultivation lineages, so these ones here, and they change along the generations in cultivation. The effect sizes for biomass, height, and number of inflorescences are expressed as proportion of the mean value of a given cultivation lineages. So we see the proportional change here. What we see is that most lineages do not change significantly over time in cultivation, which is a good thing. Some species change in multiple measured traits, and for example, Lychnis floscuculli changed in biomass, in number of inflorescence, and start of flowering. This means that it's almost circa flowered 1 week earlier. So this is change in some species. We can see, okay, there's a change over this few generations, but overall, not too many cultivation lineages change that much. But when we put all lineages into one statistic, we could see that there is an increase in biomass, height, and number of inflorescence over all cultivation lineages combined. So we see that there's an increase. It is again small, but there's a significant increase. We could say there's an increase in vigor of these plants. When we calculated we then calculated the coefficient of variation. It is this measure of dispersion or variability relative to the mean of the data set, meaning that a higher coefficient of variation means there's a greater spread of values, and a lower coefficient of variation means less variation, or the data points are closer together. And what we can see in this bottom line here is that in the biomass and number of inflorescences, so the flowers, and the start of flowering, the trend goes towards more uniformity. We see it in these three measured traits, a significant decrease in variability, so more uniformity. As you could see in the title of the paper I mentioned for this chapter, we mentioned the domestication syndrome. So I want to cover some signs of domestication, but first, really domesticated plants that have been domesticated for really long periods of time, or that are carefully bred and selected, show a decline in genetic diversity that we can see here from the study. We already know, I already showed you, that we see an increase in genetic diversity in these plants. So I'm not talking about in this few generations the plants are domesticated. We see in the talk, we saw in the talk from Walter Blecker how much work goes into preserving the genetic integrity of the plants. So these plants will not be domesticated after a couple of generations. It is more like, do we see signs or a trait shift towards this domestication syndrome? So more of a shift over all species together towards this domestication. So we're not talking about domesticated plants like these examples, but about signs or trends of the domestication syndrome. What can we see in these few generations? So some signs of domestication are increased vigor that we already saw. Uniformity, we saw this slight, significant, small trend towards uniformity with a coefficient of variation. And reduced seed shattering is a different sign for domestication. As fully domesticated plants often have seeds that stay way longer on the plant and are dependent on human harvest and replanting even. In this propagation system, only seeds that are ripe at the exact moment of harvest and are still on the plant can be collected and can therefore contribute to the gene pool of the next generation. So maybe in just a couple of generations, we could be able to see a shift or trend towards seeds that stay a little bit longer on the plant. In order to see whether we see this pattern, and because I don't value my free time, I guess, we measured the time the seed stayed on the plant, or the proportion of open to closed seed pods, like here in Lotus corniculatus. We could do that in 5 cultivation lineages. And what we find was in one cultivation lineage, only in one, seeds stayed significantly longer on the plant with increased number of generations spent in cultivation. The values are effect sizes and their confidence interval. The effect sizes are odds ratios for flowers with seeds versus flowers without seeds, meaning values greater than 1 indicating an increase in seed retention. So everything on the right of this line here. So only one is cultivation lineages is slightly significant. It is significant, but only barely significant. We see that in this cultivation lineage, the seed stayed longer on the plant, the more generations were in cultivation, the longer it was in cultivation. Other signs of domestication are loss of natural defenses or loss of ecological competitiveness. We know that domestication traits of fully domesticated plants are beneficial for agricultural use, but have generally detrimental effects on plant fitness under natural conditions. We know that from literature. And it is speculated that trade-offs between vigor and, for example, drought tolerance or herbivory resistant could potentially reduce restoration success. At least that was speculated. It's called wimpy giants. Maybe you've heard it, that the plants grow bigger, but now they are not as tough. So we looked at that in chapter 3. Do cultivated plants react differently to stress than plants grown from wild collected seeds? This is an unpublished manuscript, so unfortunately this cannot be shared, or this part has to be cut out of the upload to YouTube. Thank you very much. So what we did here is we looked at, again, the same seeds from the same seedlots and looked at the F0 seeds and the last generation in cultivation that we had. So wild versus cultivated, we call it from now on. We had 5 species, 4 treatments: drought, early cutting, competition, and nutrient deficiency. And together we had 900 plants. What we see here is an exemplary plot from Centaurea Yardsea and biomass. We found that the biomass was almost always higher in cultivated plants in all species. So we see that the cultivated plants are always a little bit bigger. But we already know that from the last study that this is this increased vigor. But it's, I guess, nice to confirm it again. We therefore applied an interaction term in the linear model, so generation times treatment, to test whether the effect of treatment differs between wild and cultivated plants. Basically, is the stress reaction different between wild and cultivated plants? Okay, I show you this table. I walk you through it. You can see here the difference between the generation, meaning wild versus cultivated. There's almost always a significant difference. Again, we already know this. This is increased vigor, old paper. Then we see that treatment is significant. Okay, this is boring as well. We know that when we give plants less nutrient, less water, then they grow not so great. But now is the interesting part: generation times treatment. The significant results indicate that stress affects wild and cultivated plants differently. That is what we want to know. For example, stress might cause a larger reduction in biomass for cultivated plants compared to wild plants. Most, all but one actually, results are not significant. This would suggest that the effect of stress is similar for both wild and cultivated plants. That is, while plants generally have higher values, the difference between them and cultivated plants remains consistent across stress conditions. Oh, and here in this one case, the Dianthus cartusonorum biomass, where we see that there's a significant difference. This could even be explained, maybe explained. It is an unfinished manuscript. Maybe we see again this increase in vigor in biomass, but we had, because we were looking at the we had this cutting treatment. We can see the cutting treatment here. We just cut them early in the growing process of the plant. We just cut them and looked how fast they can regrow. This is the cutting treatment. But what we also were able to do with this was measure the early biomass. And we could see that cultivated plants have accumulated more early biomass. So they are already bigger. What this could lead to is, in theory, need to look at the data more, that if we have the difference between wild and cultivated plant, we have already a difference when we start the treatment. So we did not start with minimal water right from the start, not to kill many young plants. So if you imagine we have still all the water in the world for the plants, and then we applied the water the drought treatment, then the cultivated plants are already bigger than the wild plants, and then they basically lock in. They don't grow much after that. And if we then measure the difference in biomass, this would be an artifact having less to do with the drought stress, but more with the already known increased vigor and faster growing. So the cultivated plants evaded our stress treatment. So in this only one case where we see a different reaction in stress, it could be explained by this already known increased vigor. Okay, so now we are back from the part that has to be cut out of YouTube, I guess. To recap this very briefly, we wanted to know, are plants in later generations genetically different? You could see this slight increase in FST values, but to give context, this increase was rather small, and we could see that the genetic diversity even increased. So yes, we saw genetic changes, but only slightly, and the genetic diversity even increases. We wanted to know if if plants are different in morphology and phenology, and we could see that only some cultivation lineages change more. Most of them don't really change that much, but over all species together, we see this slight increase in vigor and this small decrease in variation. And in chapter 3, we wanted to know if plants react differently to stress. The cultivated plants reacted differently to stress, and we could not detect substantial changes. And with that, I come to the end of this talk and want to thank you for your attention. And great thanks goes to Oliver Bosdorf, Anna Braurowa, Walter Durker, Norbert Hölzel, Christian Lampai, and Stefan Michalski, who worked on the published manuscripts, as you can see here. Thank you very much.
Thank you very much, Malte. That was really great to get all this information. Especially thank you also for sharing unpublished results, which are really important, I think, for anyone working with regional seed mixes. So, are there questions? Philip.
Hi, do you hear me?
Yeah.
Yes.
Okay. So I have a question. So you had now all these generations next to each other, and you said there are small changes in morphology and phenology. Did you maybe observe a different behavior from pollinators approaching the plant?
This is really exciting, but unfortunately, this was not part of this project or these projects. We could see, however, in the first study that I showed, where we had this kind of reciprocal transplant experiment, seeds or plants of the same species coming from different regions, put all in this one experiment in Münster, and we could see that pollinators visited the plants differently. So different communities from different regions visited the plants in a different way. So this was mainly driven by a different flowering time from the different regions. Yeah. But on this experiments on the rooftop of the university, this was not included. We didn't look at it. Yeah.
All right. Okay. Yeah. So, Philip, still some things to find out for you.
Yes.
More questions? So maybe I can ask something, because I think I understood that right, that if you find out that there is no difference in the stress reaction, this actually means that if you bring these plants back to the natural habitat after 5 generations in cultivation, there should be no problem. They should have the same fitness as the wild plants, right?
This is a really important question that needs to be answered. There are more and more studies from Teresa Klein-Rauf-Hake or Anna Lampai-Bucharova that look into this, specifically what happens with seeds that are brought back to a restoration site, so brought back to the natural environment, if you will. This was not the scope of this project. So what we can say is, just by looking at the data of, okay, we see the changes only minor. Okay, and the reaction to stress is more or less is the same. But maybe there are factors that we can't see because nature is more complex than this. As I try to show, this is one case where it might be the case in the stress reaction paper that the cultivated plants were already bigger when we applied the drought treatment, and therefore they evaded the stress. So we see that there's a bigger difference in the drought treatment, and therefore the model shows that there's a different stress reaction. But it is only because they are already bigger. So because they have more vigor and they grow faster, they evade the stress. So it could be that they react differently to some different challenges that we can't all throw at them in the experiment, other experimental conditions. So we would need to look into the real world scenarios. But what I can say is, from looking into all of these experiments and all of this what we have done, I would strongly say that the seeds are really good for restoration. They are more or less the same. There's, as Walter Bleker told, so much research and brain power going into how do you manage to do the seed sourcing system, what all you have to keep in mind, and then as a result, we see that plants do not change that much. So I would say you can use them for restoration. It's a good system.
Right. I mean, if they are larger, if they have more inflorescences, more biomass, I mean, they may attract more herbivores and more pollinators. So
of course, but again, the changes that we see are small. So it is maybe really exciting from a perspective of an evolutionary biologist to see even this trends toward domestication syndrome, with the seed retention and so on. But we are way far away from domesticated plants or plants that are way bigger. If I looked at the plots of the experimental site, the rooftop in the university, we couldn't see the difference between wild and cultivated plants. There's no way you could see the difference. Only if you gather all the data, measure it, and then look at the calculations, you see, this is cultivated, this is wild. But you can't see it if you stand in front of it. This giant must be a cultivated plant. It's not that big.
yeah. More questions.
Can I take one? Gunilla?
Yeah.
Yeah?
Yeah, go ahead, Gunilla.
Yes, thank you for your presentation. As I'm not a biologist, I am curious about what is the time frame for bringing up 5 generations of those wild seed flowers? Is it within one season or is it within 5 years? Just to explain a little bit more, if you want to. Thanks.
Of course. So I'm not sure if I understand the question, but so in this experiment, we had this old seed and the newer seeds and grew them all at the same time. So it's one growing season where we had all the seeds. But I think you meant, did you mean that?
So you had all those 5 generations grown up at the same time. You saved seeds from generation 0 and 1 and so on. Okay. So
exactly, that was the great work of the Saatenzeller and Gumpenstein that provided us with these seeds. So these seed producers are really careful and they know what they're doing. And with every seed collection, they put some seeds aside. So they gave us these really old, in part really old F0 seeds, really old F1 seeds, because they don't always use they don't use 5 years for 5 generations. They establish a field with F0 seeds, and then after a couple of years, maybe, they have to replant the field using the old seeds, the F0 seeds or as F1 seeds. So this F0 seeds could be really old. The F1 seeds could be old. And we were lucky enough to get these seeds from the seed producers and then just grow them the same year, same condition, side by side.
Okay. Thank you. I thought you collected them one after another and made and measured one generation. Each up.
That would be cool,
thank
but
you.
Lots of work. Yeah.
Okay, thank you.
You're welcome.
Now, of course, then the question is, those were all plant species which flower after the first year?
Most certainly, yeah. We have a couple of species that can flower in the first year, but more or less flower in the second year. It is a couple of years back, but I think Centaurea scabiosa may be one of them that can flower in the first year, but most of the plants will start flowering in the second year. So we excluded this from the analysis when we looked at it, that when we knew this species will not flower, then it is pointless to look at the onset of flowering where we see only 2 or 3 individuals flowered. Yeah. Yes.
So there may be some differences depending on the generation time of the species as well.
Exactly. Yeah. Of course. Yeah. Yeah, it is really hard. We try to look at different traits. I showed only the self-compatible or self-incompatible, but I don't know. At one point, I had a list with 20 different traits that could potentially make a difference in how fast species change or adapt or experience bottlenecks. But if you subset your experimental data into, okay, now we have not self-compatible, incompatible, but we have maybe a trait that has 5 or 6 subsets, then it gets more and more tricky to make a good statistic. So yes, it would be really interesting to see why some plants change, what is the difference if they have a different generation times, but with a controlled experiment, it gets really tricky after a while.
Yeah, there is a lot of diversity, so that's
yes.
You cannot control all the different traits.
Yes, exactly.
Good. Yeah, if you have a question, please go ahead. But it looks like Malte has answered all questions and given a great overview of what happens to these plants in cultivation. Walter Bleker already mentioned that they only cultivate them up to the 5 generations. So that's probably also already some feedback from Malte to Saatenzeller.
Yeah, they already, before our studies, they only made it for 5 generations. And we see that it is really good. with the 5 generations, because we see that the fixation index, the FST, increases linearly, and we see also other traits that increase linearly. Again, this increase was significant, but really small. But if you compound this over generation and generation, then maybe it would show a difference that would be biologically significant. But at the moment, we see small changes that are evolutionarily interesting, but I would say they are still really good for restoration. Yeah, so this 5 generation limit is I think really good.
Right. Great final words, giving confidence in regional seed mixes. And yeah, our plan is not just to put these videos up to YouTube, but also to write a review on the whole 6 webinars together in a way that is comprehensible to non-biologists and will help communities and companies, private persons do the right thing with regional seed mixes across Europe. And so check into our website of the EXPBIO project from time to time. And also, if you're interested in more webinars, there is another webinar planned for next winter, spring. So yeah, stay tuned to the EXPBIO project and its results. Thanks a lot for joining this webinar. Thanks a lot, Malte, again, for your contribution and also for the discussion.
Thank you very much for the invitation.